My dad was great at bringing home technology that he and his colleagues were developing, and actually set up an electrical lab in our basement. And my brother and I and sisters got exposed to a lot of different sciences. When I was 7 years old, his dad, my grandfather, lost his vision. And then my mom's dad, several years later, had a series of strokes where he became hemiplegic. Whilst I saw my dad and his colleagues creating amazing tech to shoot stuff into the sky and shoot stuff out of the sky, I saw little of anything being done to help these two guys who I cared for very much to restore function they had lost due to their illnesses and injury. And I became intrigued and motivated to think about how I could help develop technology that could enhance human health and specifically restore function to those who lost it as a result of an injury or disease or aging. Our effort that helped launch synthetic biology was interesting. This was done with Tim Gardner, my first graduate student in what became synthetic biology. And Tim and I were intrigued, what could we do in molecular biology as engineers and physicists? There was emerging interest in doing systems biology at the time. So could you reverse engineer natural networks? On the heels of the amazing output coming from the Human Genome Project, as different groups were beginning to really identify the components of our genome, components of our cells, on a large scale, in a high throughput manner. The excitement then became, could you put those together in networks and pathways? And Tim and I initially considered that problem, but the data were not yet available. So the microarray technology, an older technology, really no longer used technology, had just appeared where one could query and measure the output of multiple genes inside a single cell simultaneously. But they were not yet available at a public level where a group like ours, which didn't have a wet lab or any money in this space, could reasonably begin to reverse engineer natural networks. Most engineers are tinkerers. They put stuff together, and that's really the dominant of the engineering strands. And Tim and I got excited, well, could we put molecular components together into circuits? And we began thinking about what would be interesting to build, but simple enough that we could build it and analyze it. And we came up with the idea of modeling and designing a genetic toggle switch. This was a simple system that we designed as being made up of two genes that wanted to be on but were arranged in a mutually inhibitory network that could then exist in one of two stable states. Gene 1 is on, Gene 2 is off, or Gene 2 is on, Gene 1 is off. And you could flip it between those states. And then we did mathematical modeling to start. And it's interesting to reflect back, this was now in the late 90s, that we were motivated to build it primarily to validate our mathematical model. Most of the effort really focused on the model as the end, and few of any experiments were being done. But in the early 90s, a number of groups in the neurosciences began to look at very small systems of interacting neurons in stomatic ganglion and lamprey spinal cord, efforts done by my colleague at BU, Nancy Capel, and Eve Marder at Brandeis, Avis Cohen at Cornell, and Bart Ehrmantraut at University of Pittsburgh. And it was really fascinating to see how they had this interplay between modeling and experiment. And Tim and I thought we could do this in molecular biology. And so we teamed up with Charles Cantor, who had a wet lab at BU, and we actually began building toggle switches. And in a matter of a small number of months, Tim was able to successfully build a bistable toggle switch. It was really a flashpoint for physicists, engineers, mathematicians wanting to move into molecular biology, kind of giving them an invitation to come in, as well as a tool set and entry points. And these pieces ended up launching the field of synthetic biology, which really was not our intent. Our intent was just to do interesting, cool work that maybe we could build upon, but it had a much bigger impact than we had anticipated and or planned. 25 years on, I'm really proud of what the field has done in academia, and increasingly excited about what can be translated out of the field into different aspects of society. There are now several hundred labs around the world. Many, many major universities have research centers, programs, institutes, and there have been hundreds of companies that have launched. So it's interesting, and yet I still think it's early. We have a lot to do, and one of the more exciting areas is this really emergence now of use of artificial intelligence with synthetic biology to create what's being called generative biology. So it's kind of viewed as synthetic biology 2.0. But it's fun to see. I think it's a very exciting field for young people, where I think they're really intrigued as to how can you harness biology as technology to address some of the world's biggest problems. And I think when we look back on this century, two of the dominant areas are going to be artificial intelligence and synthetic biology.